training with Alain Bessis at EBENS, and they were a pioneer in unraveling that the mutation in a microglial gene, CARAP-DAP12, is sufficient to alter synaptogenesis. She then joined the lab of Luc Marotteau and plunged into the world of serotonin, and she's now a professor at Sorbonne University, where she leads her lab since 2019. Her research aims to understand how microglia and the serotoninergic system interact during brain development and how disruption in this communication could be at the root of some psychiatric disorders and more generally of atypical neurodevelopment. And the floor is yours, or actually the transmission is yours, please.
Yes. Okay, so hello everyone. So first of all, I'm really sorry my camera doesn't work. It's first time that it does that. Okay, I will share my screen. Okay, great. So first of all, I want to thank you very much. Thank the organizer very much for inviting me. I'm very happy to be able to share our work in the lab with you today. And so I will start. I mean, as you were connected here, I guess you know already about this the main feature of microglia, which are that the cell are very motile cells. Sorry, I'm a bit slow because I'm fighting with the screen here. Sorry. Sorry, sorry, I'm going to stop it. Okay. Okay, so these cells are very motile, and as you know, they play multiple roles throughout life. Really sorry. Sorry, I'm trying to get rid of the picture. Okay, of the participants, but I will try to do with them. Okay, so they play multiple roles throughout life, and what we are most interested in the lab is how microglia interact with neurons to regulate the formation of circuits, their refinement, but also synaptic plasticity and activity and neuronal activity. Okay. I'm really sorry. I'm going to change something because I just... Yes, okay, sorry. Okay, so let's start again. So I'm really interested in how microglia interact with neurons and their roles in circuits formation and refinement, and also how they may regulate synaptic plasticity and the activity of neurons. And so this part of the scheme. And but as I said before, so they play multiple roles throughout life. Microglia play multiple roles throughout life, but of course, all these roles are not. I mean, they are specific roles associated with specific ages of development. And the changes from, changes from one role to one function to another throughout development, it's matched with changes in, it has been shown by many groups. And for example, as illustrated here with this beautiful article by Schwartz and Amit, that microglia through development go through different programs, and many groups have shown that there is a heterogeneity of transcriptomic signature in microglia throughout development. And what we're more interested in the lab is what happens in the postnatal period. And it's very important to understand how microglia mature in the postnatal period, because this maturation of microglia is matched with the maturation of neuronal circuits, that is the formation of circuits, but also their maturation and the synaptic refinement. And thus, alteration of the maturation of microglia in this period, or alteration of their function, will impact on the formation of brain circuits, and thereby it can increase the risk for atypical neurodevelopment and disorders such as autism, schizophrenia, or anxiety disorders. And thus, it's very important to understand what are the signals which control microglia maturation in this postnatal period, but also, or more generally, microglia function in this postnatal period. And because it's a time of circuit formation, it makes sense to think that possibly neurotransmitter or neuromodulator could play a role in this maturation. And in the lab, we were interested in the possible role of serotonin. So why serotonin? Why is a neuromodulator serotonin? Well, one reason is that the maturation of the serotonergic circuits starts very early in development during prenatal development. And as you can see, at the time where you have circuits formation and refinement, it's also a moment where there is already lots of serotonergic innervation. Also, it's still ongoing. The refinement of the serotonergic system itself is still ongoing, but it develops quite early, for example, in comparison with the dopaminergic system. And moreover, the serotonin is secreted in the brain, is secreted by a few neurons which are in the Raphe nuclei in the brainstem. But these few neurons really innervate the whole parenchyma, and there is a very dense innervation of serotonin, as it is exemplified here, meaning that potentially it's possible for serotonin to act possibly on any microglia which are themselves disseminated through the parenchyma. Moreover, serotonin, it is released. It's released through. It's stored in vesicles, which are themselves stored in varicosities along axons, and it has been shown since for a very long time that serotonin release can occur from varicosities which are not opposed to synaptic junctions. And it is admitted that serotonin acts by volume diffusion, meaning that it can act at a micrometer range. And since electron microscopy studies that shown that, it had been proposed that serotonin could act on glial cells, not necessarily postsynaptic neurons, but also glial cells. At this time, they were more talking about astrocytes, but microglia could be targeted by serotonin as well. And another reason to think that serotonin might play a role in the regulation and maturation of microglia is that besides the role of serotonin in many physiological functions in adulthood and regulation of many behavior, it's also known for a developmental role since years. And many studies have shown that in case of perturbation of the serotonergic levels, and notably during the early postnatal period, you can have many defects. And when I say altered serotonin levels, there are lots of experiments where people have tried to block the serotonin reuptake by using, for example, antidepressants, with SSRI antidepressants, which act by blocking the reuptake of serotonin, or modification of the activity of the enzymes which degrade serotonin, but also studies based on polymorphisms of the serotonin transporter, which will also change the rate of reuptake of serotonin and thereby the extracellular level of serotonin, but also mutation in the enzyme, mutation in receptors for serotonin, also pretembers. All these factors will alter serotonin levels. And it has been shown by many studies that this impact on behavior by changing the anxiety, propensity to depression and aggressivity. And this has been also correlated with alteration in neuronal circuits. And all these experiments are mainly in mice, but in humans also, there are links between changing the serotonin level and or signaling and neurodevelopmental disorders, and notably the increased blood serotonin level as one of the first biomarkers for autism, and it is present in about 1/4 of the autistic patients. And thus we wanted to ask whether microglia could be regulated by serotonin, or it's another way to say it, could they be an effector of serotonin during development? But to do so, of course, microglia need to have receptors for serotonin. And when we started this study, there was already one study published on microglia or brain macrophages showing that they were expressing the 5-HT2B subtype. So in serotonin, there are about 14, 15 genes encoding serotonin receptors belonging to different families. And so we looked at first in a primary microglia cultures, and we found that the main receptors for serotonin in microglia, at least in culture, was the 5-HT2B subtype, which is coupled to GQ. I want to say that also in macrophages, serotonin receptors is expressed, and notably the 5-HT2B and 7. So it was not very surprising to find this specific subunit, subtype of receptors in the microglia. Interestingly, this receptor has a low expression level in the brain in general, and here it's the PCR done with the same primers but with a whole brain extract, and you can see that the 5-HT2B in comparison with other as the band was very very low. I must say that microglia express the 5-HT2B receptors for serotonin, and this receptor has a low expression level in general in the brain. Nevertheless, it is expressed in some neurons, and notably in serotonergic neurons. We then wanted to quantify this expression over time, this time in a quantitative way, and we observed that, interestingly, the expression level of the 5-HT2B receptor gene in microglia, I say microglia, actually it's brain macrophages, because it's purified by the microglia or cells purified based on expression of CD11B. In this case, we have we observe an increase in the expression of the HTR2B gene over development in the first 2 postnatal weeks. And just before to move on, I want to stress on the fact that it is also expressed in human microglia. As you can see here, it's taken from a single cell RNA-seq assay from a postmortem brain, and the HTR2B gene, it has globally, let's say, a low level of expression, but where it is expressed, it's mainly in the microglial cells in comparison with other brain cells. Okay, so microglia or brain macrophages do express serotonin receptors, and then are they in the proximity to a serotonergic viscosity? So the answer is yes. As soon as P2, we could see by confocal microscopy that it's not difficult to find places where microglia are really very close to a serotonergic axon, serotonergic varicosities. So as you can see, the axons of serotonergic neurons, they have a typical morphology with varicosities, as you can see here, and sometimes you see the varicosities very close to a microglia. We did at the beginning a few images at different ages, and now we are moving to a more systematic study and quantification of the vicinity of proximity quantification of the proximity of microglia and serotonergic varicosities. And so we'll reconstruct that stack from that confocal stack and with IMARIS, and then we will reconstruct the microglia and the varicosities, and then we can select or focus only on the varicosities which are very close to the microglia. And here we are starting, this is the work of a PhD student who is starting a PhD now. She's starting by quantifying the number of varicosities surrounding microglia around ages, and the preliminary results show a great increase during the first 2 postnatal weeks. So suggesting that microglia might be more responsive to, increasingly responsive to or sensitive or regulated by serotonin during this postnatal period. And of course, we quantify that now in different regions at more ages, and also see if there is a sex effect. Okay, so microglia do express receptors for serotonin. They are close to serotonin, but as I said, the receptor has a low expression level. So is it does serotonin do something on microglia? Can we prove that this receptor are functional? So to answer this question, we started by performing 2-photon experiments where we on actual brain slices from mice expressing GFP in microglia, and then we just applied locally serotonin. And as you can see here, as you will see here, yes. Okay. When you apply serotonin onto these slices, you can induce a very nice oriented growth of the microglial processes towards the source of serotonin. And a former PhD student, Fanny Etienne, repeated these experiments in wild-type mice and in mice that I will present just after, where we invalidate the HTR2B gene, specifically in brain macrophages, and you can see that this is enough to completely block the response to serotonin, meaning that the microglial morphological response to serotonin requires the expression of the presence of 5-HT2B receptor in microglia. And thus, in this context, serotonin has an attraction effect on microglial processes. To be honest, we don't know yet what this is. It's a proof that microglia do respond to serotonin. For the moment, we don't know exactly what is the role of this attraction. It could be important maybe in pathological situations, but for the moment, we don't know exactly what is the role of this attraction. Okay, and then we wanted to know whether this attraction, this control of microglia by serotonin was important in a physiological, for development or different contexts. And thus we decided to knock out this gene encoding 5-HT2B receptor in microglia to see what was the impact in the impact. And I will going to present two studies. One where we focused on the response to a peripheral inflammation, and another one where we focus more on the neurodevelopment and innate behaviors. So in what comes next, I'm going to use different models, different genetic tools that we use to invalidate a HTR2B gene, and that we could use thanks to this tool had been developed by my former colleague Luc Maroto. And so the three kind of mice I'm going to use in the slides next slide are either the full knockout for HTR2B or conditional knockout, where by crossing HTR2B flux mice with 6-1 CREAT2 mice from Wenbio Gan lab, we could invalidate HTR2B specifically in brain macrophages. And this, because it's a CREAT2 mice, we could induce this invalidation, this ablation, either since birth or after P30 by choosing the time of a tamoxifen injection. So in the slides, you will see that usually I write microglia, but it's true that it's more generally an invalidation in brain macrophages, not only microglia. Okay, so in the first part was is most of the work as we done my colleague researcher Catherine Béchade with a researcher in the team. And so the story started because we had observed in a previous experiment that in culture, primary cultures of microglia devoid of HTR2B, so microglia culture performed from H2B knockout mice, we had observed that in vitro, these microglia were over-responsive to LPS with an increased response, induced expression of different cytokines. And thus we were interested in knowing what would be the impact in vivo. And to answer this question, we took mice either wild type or at the beginning full knockout for HTR2B, injected them with a lipopolysaccharide, with a mild dose of lipopolysaccharide, and looked at the sickness behavior and neuroinflammation. So first, we observed that the lack of HTR2B, it's a full knockout, induces exacerbated the weight loss after LPS injection, as you can see here. And when we looked at we also performed 24 hours after the injection, we purified microglia and looked at the expression of different markers at their surface by flow cytometry. And normally in wild-type mice at 24 hours, you can basically see nothing special in microglia. I mean, they don't have, they barely overexpress these inflammatory markers. But in the mutant microglia, we could see, in the mutant mice, macula from mutant mice, we could see that the markers were still dysregulated at 24 hours. And so yes, it's here. And then more generally, we looked at neuroinflammation, and to this aim, we purified, we took out the brain at 4 or 24 hours after the LPS injection, or 24 hours after injection of a saline solution. And then we performed different qPCR for different inflammatory markers. And I will not go through all of them, but what the main picture, as it is exemplified here, is that at 4 hours, it seems that there was no really difference between wild type and full knockout mice, but at 24 hours, there was like an overexpression or overresponse of the different markers in the mutant mice. And to try to have a global picture of these different species, we did just a PCA in order to see how the different mice having, depending of the genotype and the treatment, how they were segregating. And we could see, as you can see here, it was quite striking to see that at 4 hours after LPS injection, both wild type and knockout mice seem to be well separated from the groups with saline injected mice. But at 24 hours after the LPS injection, only the KO mice were still separated from this group, whereas the wild-type mice that had been injected with LPS 24 hours before seemed to be very similar to the wild-type, to the mice injected with a saline solution. This can also be seen on a hierarchical clustering, where mainly, basically, there were like the mice were segregating into 2 groups. One which was with the saline injected groups and the wild type injected with LPS 24 hours later, 24 hours after LPS injection. And the other group, which was containing the 4 hours after LPS and 24 after LPS, but only for the KO, indicating that the KO mice had an exacerbated neuroinflammation. 24 hours, like a delay in coming back to a normal situation. And thus, but this was in the full knockout, full HTR2B knockout mice, and thus then we wanted to ask whether this was really due to a lack of HTR2B in the brain macrophages. To answer this question, we repeated the experiment in mice that were knocked out for HTR2B, but specifically in the brain macrophages, either since birth or after P30, in order to discriminate between a possible developmental or acute effect. And here we observed in the mice that had been invalidated since birth for HTR2B in microglia, we observed that we still had, we could mimic the phenotype of the full knockout, meaning that there was, as you can see here, increased weight loss after LPS injection in comparison to the wild type. And also, in terms of neuroinflammation, we still had a segregation of the knockout plus conditional knockout plus LPS versus wild type plus LPS or saline mice. In contrast, in mice that had been invalidated for HTR2B in brain macrophages at P30, adulthood, we had no effect neither on weight loss nor on neuroinflammation, meaning that both wild type and knockout, conditional knockout, were behaving the same in terms of neuroinflammation. So from this first study, we could conclude that the microglial 5-HT2B receptor seems to be required in the neonatal period, meaning between P0 and P30, to prevent an exacerbated response to inflammation in adulthood. And the model is the following, is that when microglia do not sense serotonin in this kind of critical period from P1 to P30, they might be primed and overreact to a second hit, which is here LPS, leading to a maladaptive response and exacerbated sickness response. But then if microglia are primed, or kind of primed due to lack of serotonin, we could imagine that they may also have an impact, do not do properly their job during maturation of postnatal circuits. And then we decided to look at what was happening in terms of brain circuits maturation in these mice. And this is a work of two postdocs in the lab, Giulia Albertini and Ivana D'Andrea. And this second part has been published this year. And so in this study, we again invalidated HTR2B gene in brain macrophages at birth. And then we focused really on the early postnatal period, that is P15, at the time where there is the maturation of microglia and maturation of brain circuits. And we first look at the morphology of microglia and observe that in the absence of serotonin instruction, the microglia had abnormal morphology with a reduced ramification and also a reduction in their lysosomal content. And because of this alteration of microglia, we ask whether this, we wonder whether this might have impacted also the maturation of brain circuits. And thus we looked at the frequency of mini EPSCs in the hippocampus and observed that it was increased in the knockout mice. And this was correlated also with an increased density of dendritic protrusions, so meaning that when microglia do not sense serotonin since birth, the neurons develop with more, at P15 at least, they have more protrusions and also more synapses. And the protrusion, not only they are more dense, but also they have an increased length, which can be assimilated, understood as a feature maybe of immaturity. And of course, it's tempting to make a link between this lack of, increased density of spines and maybe a decreased phagolysosomal compartment. So it's not possible to say whether the two are really related or causal, but at least, because the question was asked several times, we looked at at the content of presynaptic material in microglia, and we observed that it was smaller. In the microglia, there were a decreased amount of VGLUT1 in the mutant microglia versus in the wild-type context. And more recently, we started performing a flow cytometry experiment to quantify the VGLUT1 content in microglia at P15. So this has been done only on the wild-type heterozygous and full knockout mice, but as you can see, there is clearly a decrease in the VGLUT1 content in microglia which don't have 5-HT2B receptors. Okay, sorry, I see there is a question. Yeah. Okay. Okay, so what I presented just before was on the density of spine. It was in the hippocampus. We had similar result in the prefrontal cortex. But we also looked at another region which is very famous for postnatal maturation, which is the thalamus, the DLGN, the dorsolateral geniculate nucleus, where it is known that it undergoes a postnatal maturation. And importantly, it is also known that this postnatal maturation is regulated both by serotonin and by microglia. So logically, we asked whether it was altered when microglia were not responding to serotonin. And actually, it is the case. As you can see here, when in mice where microglia do not sense serotonin, you can see that there is a delay, I mean, a lack, a defect in the IPSI and contralateral segregation in the visual thalamus. So the defects in the maturation circuits is not only in hippocampus and prefrontal cortex, but also in the thalamus. Okay, so and then so we observe this defect in maturation of brain circuits, and as I said earlier, the maturation of brain circuits, it's very important to regulate development, and it altered maturation of brain circuits can be a risk factor for neurodevelopmental psychiatric disorders or atypical neurodevelopment. And thus, we, and maybe also because also of the increased density of spines, we saw that the mice may have behaviors that could mimic some features of autism spectrum disorders. And thus we assess different behaviors, and notably different behaviors that could match with autism spectrum disorders. And we started with looking with stereotypic behavior in a novel environment, and we measured the self-grooming of mice when they were placed in the new environment, and we observed an increased mice were grooming much more when they were lacking serotonin receptors in microglia. And then we looked at sociability with two tests, one performed on the young mice, P25, just before weaning, and one when they were adults. And in both cases, we observed that the mice which had no serotonin receptor in microglia since birth had a strong reduction in sociability. And then we looked at a third aspect of innate behavior, which is flexibility. To do so, we performed first a reversal learning test where mice were trained to go for a reward in one arm of a Y maze. And after 6 days, the location of the reward was changed, and we looked whether mice were learning, how fast mice were learning that they had to change change their behavior. And as you can see here, whereas wild-type mice were changing very fast their behavior and were very able to find the reward pellet in the reversal condition, the mice which had no serotonin receptors in their brain macrophages were very, had a strong defect in changing the location where they were looking for the reward. We also performed a kind of another test for flexibility, which is for social flexibility. In this case, male mice are raised by pairs, and it is known that when you change the bedding, the day you change the bedding, the mice perform less social interaction and more aggressive behavior. And this is, as you see, is illustrated here for the wild-type mice. But as you can see here, in the mutant mice, changing the bedding had no effect, which is an indication that there was no adaptation to the change of the bedding. So with these two aspects, we could conclude that the mice had a defect in flexibility. Okay, but these mice, they were invalidated for HTR2B in microglia since birth. So it can be a developmental effect, but we cannot exclude that it is also a continuous or acute effect of the lack of HTR2B in microglia at the time when you perform the experiment. So to discriminate between these two hypotheses, we repeated the behavioral experiment in mice that had been invalidated only at P30. And in this case, I will go fast because it's all the same. So we had absolutely no effect on the behavior. So it means that really, if we to conclude from this experiment, it seems that it's really I mean, if you invalidate it in adulthood, you have no impact anymore on the innate behavior. And thus, it means that it's really a neonatal regulation of microglia by serotonin in the early postnatal period, which is required for proper brain wiring and then acquisition of normal flexibility and sociability. And so the model is the following, to sum up all that I presented. It's that the lack of serotonergic induction of microglia in the postnatal window from birth to P30, it's required to get a normal neuronal circuits refinement. And if it does not occur normally, then you can have a defect in behavior with notably changes which can be related to which are found in neurodevelopmental disorders like autism. And as I showed in the first part of my talk, this lack of instruction also seems to prime microglia and make them hyperreactive to a second hit and to second inflammatory stimulus. And thus, now the questions we want to ask in the coming months and years, and that we have started to work on, it's what are the microglia genes and function which are really regulated by serotonin in this early postnatal period, and we expect to find genes that can be related to synaptic refinement. We also want to assess the role of microglial serotonin receptors in adulthood, because we have some data showing that the regulation of microglia by serotonin could be implicated in plasticity and in memory formation. And last, the level of serotonin is regulated by drugs like antidepressant, SSRI antidepressant. And thus what we are also assessing now is our microglia regulated by SSRI antidepressant, and does this participate to the effect of antidepressant? And thus I just want to thank all my colleagues and collaborators. So I already, I mean, the person who were involved in the work I presented are Catherine Béchade here, Ariane Farad, who is starting a PhD, Chloé Lorenzo, who started the phagocytosis experiment, and my former lab members, so Luc Marotto, who is now retired, 2 former postdoc and former PhD student. My collaborators, Nicolas Gervasi, who helped us at the beginning with 2 photon experiments. Corentin Lemagresse, we are 2 PI leading the lab, and Corentin Lemagresse is here, and he helped us for the, I mean, he performed the electrophysiology experiment, and also Alexandra Repsam, with whom we performed the experiments on the maturation in the visual thalamus. And then I will thank you for your attention.
Thank you so much, Sean, for this very interesting talk. So Marta will take care of the Q&A.
Yes. So, well, everyone, if you have any questions, you can either write your name in the chat and we will unmute you, or you can write the question and I will read it for you. So we already have the first question by Carlos Matute that says, are serotonin positive varicosity contacts with microglia synaptic-like?
Are they synaptic-like? It means, I mean, it's difficult. I mean, we should do electron microscopy studies to answer this question. So, which we have not done, but based on all the electron microscopy studies which have been performed, usually the varicosities, they are not opposed to a postsynaptic. I mean, to postsynaptic membrane. So they are always micrometer far away from other cells. So it's not really synaptic. I mean, I don't know if it's a question. And if it is about the presynaptic, in the varicosis, it's a place where you have the vesicles filled with serotonin. So it's a place of it's a site of serotonin release. But it's acting by volume diffusion, meaning that it's not going to act at 10 nanometers away or 100 nanometers away, but more at also at 1 or 2 micrometer away.
Thank you very much. So now the next one is Rosa Pelcel.
Yeah, so I can ask it myself. So thank you very much, Anne. So I was wondering if you had the chance to check the structurally speaking the fibers so during this you have this very beautiful data about the developmental window that is very critical. So do you have also evidence that structurally speaking you have this refinement of the serotonergic fibers?
That's an excellent question. Something we are going to we can address this, because actually in the same time that yeah, we will address this. We will address whether the refinement, whether there is refinement. I mean, let me start again. It is known that postnatally, I mean, there is a development of serotonergic axons, but also a reduction, notably in the hippocampus. And of course, we will want to, we have the tools to look if this decrease in innervation of serotonergic axon, whether it is the same in wild type and in the mutant mice. But for the moment, we don't know. So this would be interesting indeed to look at the effect of microglia on serotonergic fibers. What we are doing now, I mean, a way to do that, I mean, we will compare wild type and mutant mice. And we are actually assessing for the moment, the context between, I mean, the percentage of varicosities which are touched by microglia. And this is also regulated over development, and we will see if it is different in wild type and mutant mice, and also whether it is regulated by drugs which will modify microglia.
Thank you. Thanks.
And now the next question from the chat from Long Jun that says, great work. Wondering how HT2B is coupled with signaling required for pruning.
So for the moment, we haven't results yet, but we are performing the RNA. We have performed the RNA-seq from wild type and mutant microglia, and thus we will see if the genes which are dysregulated are linked with pruning. For the moment, I don't know. What we have done and are currently analyzing is impact of early postnatal fluoxetine on microglia. And well, we didn't find changes in the complement system, for example, but other things that might be interesting. But we expect, we might see, what we expect also is to find new genes that might be regulated, important for pruning.
Mm-hmm.
But no link with complement. I mean, it was we were a bit disappointed, but it's not so, I mean, not direct link with the complement system, for example.
Yeah, so then Jacob Dundee says, fantastic story. I was wondering whether you think the behavioral changes observed in the constitutive knockout mice at birth are due to defects in the formation of neuronal circuits during development, or microglia without the HT2B during development leads to microglial deficits that persist into adulthood and contribute to active learning.
Sorry, I'm
just quite long.
Yeah,
so
okay, I can see. I'm going to read it.
So if are due to defects in the formation of neuronal circuits during development, or
I think for the innate behavior that I presented, I mean, we think that I think we have the evidence to say that it's developmental, because I mean, I went a bit fast on this slide, but when we invalidated the HTR2B gene in microglia at P30 and repeated the behavioral experiments in adults, we didn't see any of the phenotypes, nor on flexibility, nor on on sociability. So for sociability and flexibility, it's really a matter of development. And I think it's microglia which act on the circuits, and it's definitively wrong. But yeah, this is for the experience, what I presented. But as I said briefly also, nevertheless, serotonin to microglia signaling in adulthood seems to be also important, because I didn't present it here, but we have evidence that it has an impact on plasticity.
Then Stacy says, thank you for the interesting work. And she's wondering whether why the sex differences with postnatal HT2B depletion. Do females potentially rely on a different receptor to compensate, or is there something else going on?
Actually, we perform. Actually, most of the defects that we saw them both in males and females. I mean, so for example, for the cognitive flexibility or for sociability, it was both in males and in females. So maybe some phenotypes were a bit less strong in females. So yeah, maybe it's a different receptor. I don't know. That would be something interesting to study. But females do also have a phenotype. So yeah, I think it's acting in both. Maybe it's a bit less strong. It's a bit less strong in female. Maybe there is some compensation, but I think it's important in both. And for the sickness behavior, the neuroinflammation, we saw it in, I mean, the genes we looked at, they were changed in males only. In females, they were not changed, but the behavior was changed. So I think that maybe it's different. Neuroinflammation might rely on different genes in female and in males.
Then Guy Brown asked, does serotonin directly increase microglial phagocytosis?
We try to perform such experiments by purifying microglia, treating them with serotonin, and we didn't see an effect. So I don't know. I don't know if it's an acute effect or if it's something which is changed by the lack of HT2B, 5HT2B receptor. Also in the paper published by Kettenman Lab in 2012, there was an effect on phagocytosis, but I mean, there were no studying the mutants, so I don't know if it was really specific. So I can't answer this question for the moment.
Then Daniel says, if microglia without the HT2B receptors are less ramified and less phagocytic, does that mean that in wild-type mice, serotonin causes microglia activation and phagocytosis?
Yeah, good point. Yeah, it's a possibility that the basal. I mean, I would guess that basal serotonin, I mean constitutive serotonin receptors in microglia do increase or positively act on microglia motility and phagocytosis, but I don't have the information, the answer yet.
Yes, so thank you very much. I think that's it. Paul says gotta go, but thanks for sharing your interesting work.
Okay. Well, I thank you very much, and I'm sorry at the beginning it was a bit difficult, but I was lost with my screen, and I'm sorry again for the camera. And thank you very much for all the questions, and happy to discuss more if you want to write me.
Thank you so much, Anna. Yeah, it was a pity we could not see you, but thank you so much.
Okay.
So see you guys next month, the 6th of February, with Luca Pelsotti Giametti. So hope to see you all there. Bye.
Okay. Bye-bye.
Bye.